concept. Now I am giving the floor to Ben Steckman, another established landmark research infrastructure, EU-OPENSCREEN, the European Research Infrastructure Consortium for Chemical Biology and Early Drug Discovery. So Ben, please go. We cannot hear you.
Okay, can you hear me okay now?
Yes, very well.
Okay, perfect. Thank you, Donato, for the kind opportunity to present EU-OPENSCREEN, the European Research Infrastructure Consortium for Chemical Biology and Early Drug Discovery here in this EPTRI Stakeholders Roundtable. Let me first introduce everybody to EU-OPENSCREEN before I highlight some areas for potential collaboration between EU-OPENSCREEN and the EPTRI community and how we could potentially contribute to the pediatric research. Here at the beginning, I'd like to point your attention to a problem that all of you are aware of, namely the high attrition rate in drug development. As you can see in the graphic on the left-hand side, the main cause for the high attrition rates in the clinical phases 2 and 3 is the lack of efficacy. So we'll not try to convince you that an individual organization or approach can solve this issue alone. So I think everybody agrees that this requires a set of approaches to addressing this multifaceted issue. And even though lack of efficacy itself has many underlying causes, it is attributed, at least in part, to the selection and the validation of the drug target. And what we observe, and others as well, is that pharmaceutical companies limit efforts in their early drug discovery, and they focus on those very few drug targets they have been thoroughly validated by using chemical probes, which are well-defined mode of action. And it's here that we see the importance of academic initiatives to develop novel chemical probes for the validation of new drug targets. And in this context, we hope that EU-OPENSCREEN will also contribute to making an impact on the development of novel drugs. Next slide, please. But it's not only the pharmaceutical industry, but it's also the academic scientists that focus on previously well-studied molecular targets. And in general, studies that introduce a novel chemical probe for a cellular target are usually published in high-impact journals and are among the most cited publications on that target. And what we observe is also that the availability of a chemical probe for a given protein strongly encourages other researchers to study this particular protein. And chemical probes and antibodies are arguably the best tools to advance research on a given cellular target. Next slide, please. And it is in this context where EU-OPENSCREEN, as a new European research infrastructure, supports researchers in the academic chemical biology to develop novel probes for target validation, etc., but also in early drug discovery to provide novel chemical matters or new leads to be starting points that can be followed up by private organizations to develop novel treatments. As a research infrastructure, EU-OPENSCREEN provides open access to technologies, for example, high-throughput screening platforms, access to resources like compound collections or public datasets, and access to expertise in high-throughput screening, assay development, data analysis, or medicinal chemistry. EU-OPENSCREEN supports scientists from both academia and industry to implement their own research projects. So we as an infrastructure are target agnostic or disease agnostic. So the expertise on the biological expertise around the target comes from the external users, and we would provide, as I mentioned, access to technologies, resources, and the expertise. EOPENSCREEN receives long-term funding from currently 8 member countries, and it was established as a not-for-profit European Research Infrastructure Consortium in April 2018, and we are based in Berlin in Germany. Next slide, please. This slide aims to kind of summarize very simply the organizational structure of EOPENSCREEN. On the top left, you see in blue, these are the components of the actual ERIC, this non-for-profit organization. And there we have a central office that facilitates open access to the resources for external researchers. We have a compound collection, a general purpose diversity collection of 100,000 commercial compounds. And this is a growing library where we add compounds that we source from the chemistry community. All the primary screening data around the bioactivities of these compounds that we screen in the various assays will be made available in our open access database. And this database also has links to already established and community recognized databases such as the ChemBull database or the PubChem database. And also training is very important for us on the one hand side for our partners to allow training of platform staff, exchange of experience and staff exchanges, but also on the other hand, training for external researchers, for example, in assay development. And as a European research infrastructure, EOPENSCREEN is a distributed infrastructure, as I mentioned, in 8 countries. Our partner sites, which can be, for example, the screening platform, but also medicinal chemistry groups, they are located across Europe in these 8 member countries. And as an infrastructure, we open access, we provide access to these resources within our network to external scientists. And we can group them in the 3 major user groups. The first are the biologists, who have the biological expertise around a target, and ideally they have already some sort of biological assay that in principle can be translated into an assay that we can use for our high throughput screening projects, with the aim to develop novel probes for their proteins of interest. The second user group are the chemists. As I mentioned, we source novel compounds from the chemistry community. The aim here is that the organic or medicinal chemists in total synthesize hundreds of thousands of compounds every year, and this in total these compounds represent a unique diversity, chemistry diversity, which in many cases is not available. It's also not available through commercial suppliers, and we as infrastructure would like to make these compounds more widely accessible for the scientific community. And the third user group are then also, as I mentioned, we make our bioactivity data available, are the users that use the database. And in the next slide, I will focus on the biology users, because I think in this context here, these are the most, let's say, relevant user group. As I mentioned, usually the assays come from outside the scientists, and we transfer these assays onto one of our screening platforms. We help them together with the biologists to miniaturize these assays, to optimize these assays until they are in a stage where they are robust enough and give a good signal, so that we can use these assays to test hundreds of thousands of these small molecules. So we then do either high content or high throughput screening by testing our own small molecule libraries using a variety of different readout technologies that are commonly applicable to the majority of assays. But because we are distributed network of up to 20 partner sites in Europe, we also have access to more specialized detection methods that are useful for individual more specialized assays. EU-OPENSCREEN has various compound libraries available. I mentioned our small molecule general purpose diversity library of 100,000 compounds. We have also smaller libraries available, which are then have more focus on that then contain known bioactives, and they could also be used for drug repurposing projects. Earlier this morning, we also heard fragment-based drug discovery. So we have also a fragment library available, and locally through our partners, we have also natural product collections available. Thanks to our automated liquid handling instruments, we also have the flexibility to accommodate different assay formats with access to various biological safety levels. And here as a last point, it's a very short, but it's the chemical hit to lead optimization, which is in principle can take several years. So it's equally important as all the above mentioned bullet points here. Next slide, please. And given the services that we in general provide to biologists, it's these areas where we can see a possible collaboration with the EPTRI community by contributing services that are of interest for the pediatric research community and that are not, let's say, at this scale available within EPTRI. And this could be access to compound collections, high throughput screening capabilities, medicinal chemistry optimization, with the ultimate goal to develop novel probes or leads that can be relevant for the EPTRI community for target validation. And EPTRI as an established community, a large community, would help us also to engage directly with the pediatric research community, and then we could advise early on the pediatric researchers on how to develop a suitable bioassays that we could use for high throughput screening. Next slide, please. So this is my last slide, just to give you an overview. I think EPTRI is in an early stage, trying to get onto the ESFRI Roadmap. We as EU-OPENSCREEN, and I think it's similar to InfoFrontier or others like BBMI, EPTRI, ELIXIR, and the others, we have kind of followed this path already. We were included in the ESFRI Roadmap in 2008. We started with the preparatory phase in November 2010, at a time when also I joined EU-OPENSCREEN. And after this 3-year preparatory phase, there was kind of a long, sometimes difficult negotiation phase with the funders in the interim phase. And as I mentioned, in April 2018, so 10 years after inclusion into the roadmap, we managed to be established as an ERIC, and we are in the middle of the construction phase, building our database, the compound management facility, and the compound collections, and we will hope to be operational and implement the first projects later this year. Thank you very much.
Thanks a lot, Ban. Very, very interesting presentation, and very nice seeing the long way from 2008, 12 years. So we are right at the beginning, but hopefully we will reach the same stage. So and we will be happy to work together, of course. And now the next presentation, there is already an example of service developed in collaboration with an existing research infrastructure, ELIXIR. This service is on pediatric data interoperability. It will be presented by Pierluigi Martelli from Bologna University on behalf of the Italian node of ELIXIR that is coordinated by the National Council for Research. So Pierluigi, please, the floor is yours.
Thank you, Donato, for the nice introduction. Can you hear me?
Yes, very well.
Thank you very much. So I'm Pierluigi Martelli, working at the University of Bologna. I'm here since I'm the Italian representative for the interoperability platform of ELIXIR. I will start presenting briefly ELIXIR and then presenting the activities that we envisage to develop in collaboration with EPTRI. ELIXIR is a research infrastructure that is devoted to the storage and analysis and all the life cycle of data in biology. Of course, biology is becoming a big data science. New technologies are are allowing to collect a lot of information, a lot of data, and this data need to be treated in a proper way in order to extract information. For that reason, ELIXIR has been established in 2016 as a landmark European research infrastructure to bring together most of the bioinformatics resources across Europe, and in order to establish some standard for databases, for softwares, for computation facilities, and also for training materials and best practices that are two of the main issues of the ELIXIR activities. So this single infrastructure is devoted to create life science data and life science in all the aspects. So it creates data concerning environment, industrial application, agriculture, and of course, medicine. Next slide, please. So ELIXIR is structured as a central hub that is located in Inkstone in UK, and involves 23 different states across Europe, and it has a hub and spoke model in which we have 23 nations and collects some 160 different organizations. Next slide, please. Next, please. And as to the scientific organization, the core of ELIXIR is organized into platforms, 5 platforms: data, compute, interoperability, tools, and training platforms that are the core of the activity of ELIXIR, of the computational activity of ELIXIR. And these platforms are in communication with user communities that collect people, scientists from different life science domains, including, for example, rare disease. And in that respect, we have ELIXIR has connection with EPTRI that was brilliantly introduced before, with human data, and with many other communities that can have a connection with EPTRI. And moreover, these communities are increasing, the panorama of the communities that are interested in collaborating with ELIXIRs and in proposing the requirements and proposing their activities to the ELIXIR platform in order to have services from the ELIXIR platform is still increasing. Next slide. The platforms, of course, represent the main activities that are required in a modern big data science. Of course, big data science means that we have a lot of data, and these data are usually heterogeneous, fragmented, are in different databases, are collected with different techniques, maybe are stored with different standards, and has to communicate or to be stored in different databases. Of course, this data need to be analyzed in order to extract biological information, and so we have different tools, many different tools that have been developed for different aims by bioinformaticians, and these tools have to be connected into complex workflows, and of course the tools have to run on a computational platform, and also computational platforms can be of different types. So we have now cloud computing, but we have also high performance computing facilities depending on the needs. And of course, only the seamless communication of all these aspects can drive to an efficient research, to an efficient extraction of information, and also to an efficient application of the knowledge. Next slide. And what is needed to make the system, the ecosystem, the computational ecosystem to work efficiently is interoperability. For that reason, the first activity of collaboration with EPTRI is about interoperability. Interoperability is the aspect of the data life cycle that concerns the definition of standard formats, the definition of standard ontologies, so the definition of standard vocabularies for referring to different concepts, the establishment and the adoption of stable identifiers for referring different objects, to have metadata to establish and to define a large and complete set of metadata that makes the data findable and make the data shareable among different researchers, so each researcher know what the data contains, and also make possible to use the data in an automatic way, because the tools will request for the data that are described with a given set of metadata. Of course, there are privacy issues, there are access issues that have to be created, and also in this case, the interoperability platform of ELIXIR is developing tools, and we need also technologies that are able to deploy the different tools on different computational architecture and to write workflow for analysis in an easy way, also for people that is not becoming from computational backgrounds. Next slide, please. So all this process is called the verification of data. We need to make our data and our tools findable, accessible, interoperable, and reusable. And this ensures the process automation because it ensures the possibility for data to be read through machines. If we use standard format, standard metadata, standard ontologies, we can automatize the process. We can have a streamlined acquisition and integration of data for their analysis, eliminating the need of changing format to make uniform the, for example, the ontologies, or to make the format uniform. And so only the verification of data ensures that we can answer scientific queries in a flexible way with a time to value significantly reduced, and with an acceleration of the research and development, also in the field of personalized medicine. Next slide. And for that reason, what we envision is to apply different services, different tools that have been developed within ELIXIR through the years that I have no time to discuss in detail, to the pediatric data. And so we think that we can start an activity for defining the most suitable standards, the most suitable ontologies and annotation for metadata, for collecting the available registers, services, or maybe to establish new services of this type, to improve search engines, to improve the findability of data by establishing services for identifier resolution, for mapping, for make the data findable from everywhere with simple searches, to harmonize tools and pipelines through workflow, and to have, in particular, also training of all the scientists that produce data for improving the interoperability of data, for make the data reusable from all the community. Next slide. Next, please. So the activity that we plan in the future is to establish a new library, an electronic library for collecting the digital documents produced that are relevant for the community and that are produced during the research activities. So repository to have a common service for data interoperability that is mainly the objective of the ELIXIR collaboration for discovering, accessing, integrating, and analyzing biological data, and also to develop tools for the automatic or semi-automatic mining of the documents, so tools also based on natural language processing for a semantic search, for a classification of the documents, and for an effective search of the information through the documents. This activity will be developed by the Italian node of ELIXIR and also from the Luxembourg node of ELIXIR at this moment. Then we hope to expand the interest to other ELIXIR nodes. Next slide. So simply to graft what are the first activities that we envision. We envision to have an exploratory phase that is needed to collect the requirements of the community. Many of the requirements will be similar to the requirements, for example, of the rare disease community, but of course any community has their specificities, so we have to investigate what are the peculiarities of the pediatric translational medicine community. So after this phase, we plan to prioritize the possible interventions. And in particular, the first intervention will be the definition of the data we have to deal with, to define the standard formats for data, for metadata, what are the ontologies that are most suited. In case ontologies are not defined, we can plan to study new ontology, to define new ontologies and new formats. Of course, we will push for the adoption of the best practices to make all the available resources findable and interoperable. So to make the available data findable from all the researchers in the community. Of course, we plan, as I said, to have training activities to train the researcher in collecting data and storing data with the best practices for interoperability. Next slide. And this is the last slide. So I was a bit quick because we are late, but for any question, I'm available also offline, as said before. Thank you very much.
Thanks a lot, Pierluigi, for describing the huge work ELIXIR and EPTRI are doing together to create this pediatric data interoperability common service. And now I have the pleasure to introduce Nada Witsianiova. She will have a presentation on the ESFRI framework to promote excellence science in Europe. And Nada knows very well EPTRI and all the work we are carrying out to create this new research infrastructure. It was a pleasure also to visit her in Prague with a preliminary proposal from EPTRI in a structured, organized meeting between new emerging projects. So thanks, Nada, for all your support so far, and the floor is yours.
Hello, everybody. Can you hear me?
Yes, very well.
Good. Thank you. So I have been a delegate of the Czech Republic in ESFRI for 7 years. I have been in the ESFRI Executive Board, so I know a little bit about ESFRI. And I must say that I am really happy that I see all the projects thriving, because in the first roadmap, EPTRI, BBMRI, InfraFrontier were included, and I hear how they are thriving and getting stronger and stronger. And then ECFIN, ELIXIR, and EU-OPENSCREEN was added in 2008, and also they are now landmarks. Wow, this is really good. Okay, next slide. So what is ESFRI? Most of you know, but maybe some of you don't. So it's a body which was formed in 2002 after the ministers decided that there is a need for a strategic, systematic approach to the area of research infrastructures in Europe. Because research infrastructures structure the European research area, and they contribute to inclusion of new member states, of associated states, and they structure the communities in which they provide services. So in 2002, the European Strategy Forum for Research Infrastructures was established. Then after the initial phase in 2004, the Competitiveness Council decided they should work on a document. So this document is a collection of projects which were evaluated to be of pan-European interest and impact. So in 2006, the first ESFRI Roadmap was launched or presented first. On the picture, you can see John Wood, the director of the Oxford Diamond Synchrotron at that time, and the Commissioner Potočnik. Next slide, please. Okay, so why do we have ESFRI strategy-led systematic approach to research infrastructures, which result in multilateral initiative leading to better development, construction, and use of research infrastructures in Europe? The main, sometimes I see that people don't understand why a research infrastructure is different from any kind of instrumentation which is in their own research organizations. The research infrastructures are very expensive, complicated, unique instruments, data sets, services, which are provided not to the home community, but mostly, or half and half, to the external users as a service. This is a big difference, and it should be in all proposals to the ESFRI roadmap very well and explicitly stated which communities this infrastructure will serve. So why is ESFRI so different from other working groups? Because it's informal. Most of the things which are negotiated in ESFRI are done in a very friendly, open way. There is also a session which is closed, a closed session where no minutes are taken, and people can speak freely their minds, and it really helps a lot. Because ESFRI is formed by a very specific combination of delegates. One delegate is always from a government institution, a ministry, or sometimes even an agency, like Britain sends STFC delegates many times. And the other one is an excellent researcher. And ESFRI is really based on excellent researchers. As I said before, John Wood, professor in Oxford, Hans Chang from Netherlands, then Carlo Rizzuto, Professor Carlo Rizzuto, Papa Rizzuto, as I call him, then John Wormsley, now director of ESS in Scandinavia, and others. ESFRI is very flexible. It can establish working groups. It establishes working groups for the road mapping process in different scientific disciplines, but it also establishes working groups for specific issues which are deemed to be important for Europe. Myself, I was head of the Regional Issues Working Group for 2 years, and we elaborated a report upon which the combination of structural funds, national money, and European money from the framework programs were proposed as a funding scheme, and this scheme was really successful, as you will see later. But other also working groups are being established for evaluation, etc. What is needed at the time, which is perfect. Then another principle is the variable geometry, which allows for member states to enter into consortia upon their national priorities or strength of their researchers community or needs of their industry, which means small member states can enter into some of the consortia and let the others do the work for them and be the users and concentrate on what is their priority, in which area they are good. Okay, so next slide, please. So I was supposed to say how ESFRI promotes the excellence of European research. I think the excellence begins even in the methods of work, which are not bureaucratic, which are open and based on reason. And what is the main outcome of this work are the roadmaps. You can see all the roadmaps. I didn't know that there will be another picture very similar, but repeating brings wisdom. So you can see that the roadmap is not a final document. It's a road mapping process. It responds to changes in the needs of the European society. And there is, as you well know, a deadline which we will meet in the beginning of September for a new roadmap. So what is important for a new proposal is to fit in the landscape, to bring new added value, European added value, and to fit in the family or in the ecosystem of already existing projects. So I congratulate EPTRI organizers that they invited many of the ESFRI landmarks in the biomedical area. Okay, next slide, please. So what are the outcomes or achievements of ESFRI? Many are of the type of policy-making documents. Sometimes for a researcher, it may seem that this is something we don't need, but we do need such a direction to see the vision of Europe and to communicate with the ministers and the policymakers and the decision makers where the money will flow and what are the bottlenecks issues we are facing. So be patient with the policy making documents and try to contribute to them, not only to the roadmaps, but only to other ones. And the ESFRI roadmaps were reflected into national ones. So for example, I was coordinating the first, second, third Czech roadmap of research infrastructures, and almost all nations of Europe have such a national roadmap. So be sure that your project is somehow included into your national roadmap. Another outcome was the process of siting of major research infrastructures. I personally was involved in the siting of the European Spallation Source, and there was the process set up by ESFRI, and we did collaborate with Katerin Cesarski and Tom Mason and others as perfect experts, and then with the ministers. And I think the process was transparent, and finally ESS was the site was chosen to be in Lund in Sweden. Another outcome is the so-called ERIC regulation, because in 2007 it was very clear that the European legal entities do not fit the needs of a European research infrastructure organization. So discussions were led on expert level, then on not so expert level, more political, with lawyers, with the Commission. And then in 2008, there was a proposal of a council regulation for a community legal framework for European research infrastructures during the French presidency. But the negotiations were not concluded until the end of the French presidency, so the Czech presidency took this task, and we finally in June had an adoption of this community legal framework for this ERIC. And we saw today that the ERICs are thriving. Now they are organized in something which is called ERIC Forum. And here on the right, you can see the map of Europe. Dark blue are the countries where the host node or the center of the ERIC is hosted, and light blue are the ones who have nodes. And we were talking about nodes. All biomedical research infrastructures are distributed. The single-sided one is, for example, this ESS I was talking about. And if you would like to see the virtual one, is the Géant network, for example. So next one, please. So when I came to the ministry in 2006, I learned European policy, as I am a chemistry engineer in the beginning of my career. I have been, and the Czech Republic entered into the European Union, and then the Czech Republic was also invited to ESFRI, and I became ESFRI delegate in 2007. And this is the situation we had, because you will know that we became free in 1989, and then our politicians and officials wanted to enter into organizations of international character like CERN, and they did quite a lot of good work, but we didn't have too much investment money. So you can see the situation in 2008. Please, next slide. And here you can see the situation in 2020, in the beginning of the year. And this is the result of work in ESFRI, a combination of European funds from framework programs, national money, national roadmaps, ESFRI roadmaps, and structural funds for investment. So, so I think it's worth it. It's worth trying to do to make a difference. And this is it. Thank you.
Thanks a lot, Nada. It was really interesting. The description of the ESFRI framework, really the most relevant instrument currently in the European research area to promote excellent science. So we are very willing to keep this pathway and bring EPTRI to the goal of becoming a research infrastructure in the ESFRI, and the vision for EPTRI to be in the ESFRI landmark will be provided with the last presentation from our scientific director, Giovanni Migliaccio. Please, Giovanni.
Hi, everyone. Compliment from my side for being able to stay so long with the history of EPTRI. May I have the first slide? The next one. Thanks to Nadia, I don't have to spend a lot of time explaining what is ESFRI, what is the roadmap, and what is this public guide. The relevant point is the request from ESFRI that the new project need to be integrated with the landmark already existing. Next slide, please. This is the process to get to the ESFRI roadmap. We already started in 2019 with the interaction with the various nodes and the ESFRI representative. And we have a deadline for September 2020 because we need to present our application to enter in officially in the ESFRI roadmap. Next slide. So what EPTRI did during this time to get in the position to apply and to interact with the existing landmarks. In slide 4, we presented the ESFRI definition of landmark RI. These are RIs which have been implemented or are operational, representing a major element of competitiveness for the era. The landmarks research infrastructure can be already operative or can be just in advanced construction phase. The listing of the landmarks you can find on the website of ESFRI. They are always increasing as ESFRI progress in the successful project of itself to create a number of structures that are relevant for the research in Europe. Next slide, please. So EPTRI did a very simple things. Let's look what is important and relevant for the activity that are in scope for our infrastructure. So during the design study, the relationship with the existing landmark RI was carefully considered. Of all the RI in the biomedical field, 8 have been identified as delivering research services in areas overlapping the scope of pediatric basic and translational science. EATRIS, ECREN, and BIMBMRI were already part of the project itself as members of the EPTRI Study Design Consortium. Infrafrontier, EU-OPENSCREEN, ELIXIR, Euro, Bioimaging, and Instructs were identified as involved in the general drug development process. Next slide, please. So we have a 3-step process. We have the survey of the landmarks, scope, and services. And then we did just that. We asked them, okay, please let us know exactly which kind of services you provide, what are the current activities, so that we can assess against what we need and the gaps that we identified for pediatric research. So we had at the end an inventory of common services and shared expertise to focus on areas where EPTRI expertise and offers would be unique. So we avoid to do and offer services that are already in place, but focus on what EPTRI has of unique to offer. Next slide. So in slide 7, it summarizes the content of the survey performing among the existing research infrastructure, which included the question to identify the relevant services for the pediatric community. These questions focused on the availability of services relevant in drug development as target identification, imaging, animal and in vitro models, pharmacology assays, biomarkers and biobanks, product development, development of biology and epidemiology. The respondents were also asked if they received requests related to pediatric research and which other services relevant to pediatric drug development can be provided at this time. Next slide. In slide 8, we summarize the results of the landmark survey. Most of the services in the drug development field are already provided by existing ERI, often by more than one of them. In fact, many services were offered in different fashion or detail from the existing landmark research infrastructure. As generalists, the landmark ERI did not track pediatric-specific request of service. So it was not possible to evaluate in detail how many pediatric researchers used the current landmark. A survey of possible users performed in the design study revealed a general lack of knowledge about the research infrastructure. So this was about, in general, research infrastructure, not about pediatrics. So still we don't know how many users were already satisfied by the existing landmarks. There are also relevant research activities and services within the EPTRI community that are not provided by any of the existing research infrastructure. In particular, the research area specific for the youngest element of the pediatric population, from newborn to toddler, were rarely addressed in the current offer. Technologies that can be offered to pediatric users by creating common activities with EPTRI have been identified. In fact, areas where novel technologies need pediatric-specific expertise to be applied to the pediatric population were identified with the possibility to update, expand, or share some service. Next slide. In slide 9, we summarize the actions taken to follow up on the results of the landmark ERI survey in the preparation of the EPTRI concept design report, the CDR. The first action was to limit the services offered or provided through EPTRI to avoid duplication. The second was to set up collaborative activities for technologies and services of common interest that need to be adapted to the specific needs of the pediatric population. Next slide. So we defined a category called the common services. We have already said, as requested by the ERIC, that all the service provided by EPTRI will be processed through a single point of access. A common service office will then manage all the requests arriving and either refer to the relevant ERI or to a bilateral multilateral collaboration for the one that are most required or requires a combination of services or require a multiple infrastructure to be provided. In addition, we need to set up some new project because not all the requests can be satisfied. So a common pathway to develop novel shared service can be created and shall be. Next slide. So EPTRI has three main options of collaboration with this landmark infrastructure. This graphical depiction of the collaborative option between EPTRI is in slide 11, and in addition to joint partnership for service providing, there is also the setting up of collaboration both at the program level and for specific projects. Next one. As action already planned in the grant that we applied, the ESFRI 02 2020, we have a lot of activities to foster the cooperation with the existing research infrastructure, and mainly to establish formal collaborative agreement with them so that we can start already to provide common services. Next one. And then I try to be short, that given the time. I thank you for your attention, for staying with us during all this long day, with the hope that the relevance of EPTRI for the future of the pediatric research in Europe was allowed to transpire in our work. Thanks again to all for your attendance.